Hello, my name is Nicole Brown, and today I will be discussing the effect of atomoxetine on depression-like cognitive and parenting disturbances that I have been investigating in the Pereira lab at UMass Amherst. Postpartum depression works in a cyclical way unless it's treated. In terms of the mom's well-being, there are hormonal and physical changes in the maternal brain as a result of having postpartum depression. In this research, we'll be focusing on the alterations of norepinephrine and dopamine and their systems. The manifestations of these changes include motivational and cognitive deficits, irritability, experiencing a lack of energy, and listlessness. These changes to mom's well-being inevitably impacts her parenting abilities as well, such as her attentiveness, her sensitivity, and the adaptability to different situations. As a result of these parenting deficits, the child's well-being is also negatively impacted in terms of social, emotional, and cognitive development. These disruptions in development even impact the child's future parenting abilities as well. Therefore, mothers with untreated postpartum depression have the potential to have generational effects. One specific cognitive deficit seen in mothers with postpartum depression is a lack in cognitive flexibility. Cognitive flexibility is defined as the ability to switch between behavioral strategies in response to changing environmental demands. So what does this mean in terms of parenting? Well, it's involved in the way a mother figures out which of the many possible reasons her baby is crying. It helps a mother adapt to the constant changing that occurs in the early years of their child's life, seeing as development occurs at a more rapid pace at these stages in life. The needs are also constantly changing of the child. It also even assists a mother in balancing all her responsibilities that she has on top of those that also come along with being a parent. In this study, we focused on the medial prefrontal cortex brain region where mood, cognition, and behavior are all regulated. This makes it an important region when discussing the brain's neuroplasticity as well. The medial prefrontal cortex has also been shown to be an integral part of cognitive flexibility and parenting, as there are significant physical changes that occur during pregnancy and the postpartum period in this brain region. These changes include alterations in the levels of monoamines, such as norepinephrine and dopamine, which have also been directly linked to postpartum depression. Additionally, in the 2008 Floresco et al. Paper, the researchers adapted a methodology to examine the role of the medial prefrontal cortex on learning and cognitive flexibility. They showed that the inactivation of this region impairs set shifting, which is changing between types of learning you use. So this shows how flexible their cognitive processes are through these trials that the paper adapted. For this study, rats were used as the animal model, including a specificity to depression as well with the Worcester-Kyoto strain. We used the well-researched and well-validated rat model of depression, Worcester-Kyoto's, because of its similar parallels to the human presentation of postpartum depression. Similarly to humans, Worcester-Kyoto moms have altered levels of norepinephrine and dopamine in the medial prefrontal cortex due to an increased density of norepinephrine transporter sites, which increases the rate of reuptake and decreases the effects of these monoamines. In the Pereira lab, we have also proven that Worcester-Kyoto moms experience similar behavioral, cognitive, and motivational deficits to human moms with postpartum depression. In order to compare the Worcester-Kyoto's results to a controlled baseline, we also used the well-researched and validated Sprague-Dawley strain as a healthy animal model. This leads us to the question, does increasing monoamines such as norepinephrine and dopamine in the medial prefrontal cortex ameliorate cognitive flexibility deficits in Worcester-Kyoto moms? So for the methods of the study, it is an adapted version of Floresco et al.'s methodology in their 2008 paper that measured cognitive flexibility. In this paper, it was shown that the medial prefrontal cortex impacted the performance and result of these tasks. In order to increase the levels of norepinephrine and dopamine in the medial prefrontal cortex, atomoxetine hydrochloride was used. It is currently used to treat ADHD and it is a non-stimulant, so there is evidence of atomoxetine's therapeutic and non-addictive potential. It functions as a selective norepinephrine transport inhibitor, which means that it causes the norepinephrine to stay in these synapses longer, hopefully counteracting the changes in norepinephrine that is seen in postpartum depression. Control and experimental groups were also utilized in this experiment to examine the specific cognitive effects during postpartum depression. These were broken down by whether the mom was a Sprague Dolly or Worcester-Kyoto rat, whether she was tested during her virgin or postpartum period, and whether she was giving saline or atomoxetine. So the first step in this methodology is to pre-train the rats. Through these trainings, the rats were familiarized with the aspects of the experiment that are not so relevant to the results, but are still necessary for them to acclimate to, such as simply being in the operant chambers and the process of inserting the levers into the chambers, as well as associating the lever with getting a food pellet. The training is concluded with a side bias test, where it is determined if the rats have any preference to the left or the right side lever. For the groups that are tested postpartum, the rats were given some time to mate and give birth, where then 6 days after giving birth, the mom goes through a discrimination training. The first test is the visual cue discrimination, where, as shown in this left picture, the correct answer for the mom to press will always correspond with the illumination of the light on the same side. Once criterion is met for the visual cue discrimination, the rat's learning technique is now changed to a response-based strategy. Now, the correct response will always be left or always be right, regardless of the position of the illuminated light, as depicted in the image on the right. During this initial response discrimination, the correct side is the side opposite of the one determined in the pre-training side bias. For example, rat mom A preferred the left side during pre-training, so during postpartum day 6, this response discrimination, the correct answer will be always right. On the last day of trials, postpartum day 7, the rats go through more response discriminations, but now they're alternating which side is deemed correct once criterion is met each time. So continuing our example, mom A would start this day on the correct answer being always left, and then it would alternate for a determined number of rounds, so always left, then always right, always left, etc. Then in the experimental groups, the rats were injected with either saline or atomoxetine in their body cavity intraperitoneally, and then performed the last discrimination. This is the extra dimensional shift, which is the final shift to a visual cue. So calling back to that first discrimination in the series, the correct answer would now be based on the location of the illuminated light again. So if atomoxetine increases the norepinephrine levels sufficiently, it is expected that atomoxetine would also elevate these cognitive deficits. This would show itself as a quicker shift to this final visual cue discrimination in comparison to untreated Worcester-Kyoto moms. So getting into our results, this first graph shows the strain difference between our 4 main discriminations throughout the experiment. As you can see, on average, Worcester-Kyoto rats need more trials than the Sprague Dolly rats to reach criterion, which is the threshold to have learned each discrimination. This further evidences that the Worcester-Kyotos exhibit depression-like behaviors, which are impacting their learning abilities and cognitive flexibility. This graph now shows the 4 groups, adding the atomoxetine-treated rats as well. Because we are only solely interested in the effects after atomoxetine administration, we'll be focusing on this last extra-dimensional shift, as the 3 discrimination trials prior are all completed untreated. So on average, both Sprague Dolly and Worcester-Kyoto groups treated with atomoxetine had to complete more trials in order to reach criterion than the saline groups. After atomoxetine injection, there was a lot of variability in the total trials needed to reach criterion. There were some rats that were given atomoxetine who Worcester-Kyoto specifically too, rats given atomoxetine who could complete the last visual cue discrimination in under 150 trials, whereas some of the other Worcester-Kyotos took over 250. So there was a wide range in these subgroups, which is indicated by the error bars. This graph shows the number of errors made in each of the discriminations, solely reflecting the two strains right now. So in terms of errors as well, Worcester-Kyoto rats make more than Sprague-Dawley rats, which again shows that there are initial differences between the two strains and further emphasizing that the depression model of rats, Worcester-Kyotos, struggle more with these cognitive tasks. Finally, this graph compares the number of errors made across the four groups. Again, we want to focus on the extra-dimensional shift as the three discrimination trials prior are all non-treated, non-saline, so they're relatively baseline figures. Atomoxetine-treated rats within both the Sprague-Dawley and Worcester-Kyoto strains again made more errors on average and again had considerable variability within their groups, which follows, seeing as the errors are based off the number of trials that they complete. So through these tasks, we are able to confirm that Worcester-Kyoto mothers show deficits in cognitive flexibility, which is congruent with mothers also affected with postpartum depression. We also see that once the rats were treated with atomoxetine, there is a bimodal effect where the cognitive deficits were ameliorated in some, but also exacerbated in others. This distribution was seen in both Sprague-Dawley and Worcester-Kyoto rats who were given atomoxetine. For further research, it would be beneficial to group Worcester-Kyoto moms based on their severity of depression-like symptoms. Like humans, Worcester-Kyoto depression-like symptoms can be variable based on the rat. Because of this bimodal distribution on the extra-dimensional shift, it is possible that atomoxetine at this dosage is beneficial dependent on the severity of symptoms. Also, including additional doses of atomoxetine in the study could help us better understand the relationship between the dosage received and the severity of depression-like symptoms. This is valuable in order to maximize therapeutic potential of atomoxetine and ameliorate the deficits in cognitive flexibility and therefore parenting abilities. Thank you for your time.